Hello, and welcome to our webinar about next-generation protein sequencing and its power to shape the future of in vitro diagnostics. I'm Ian Rogers. Today, we're so pleased to be joined by our clients at the Karolinska Instutet and talk about their work in Alzheimer's disease and how next-generation protein sequencing impacted. Our first stop today will be a brief introduction into the changing landscape of in vitro diagnostics and the new demands placed on IVD researchers and companies around the world. Next, we'll talk about how next-generation protein sequencing and proteomics in general can be fit into the in vitro diagnostic pipeline to enhance discovery, development, and approvals. It will then be my pleasure to hand it over to our guest speaker to hear about immunoassay development for Alzheimer's disease, and then to hear from Monique Seymour, my colleague, about what Rapid Novor is up to in the world, decoding immunity to enhance human health, how next-generation protein sequencing works, and some of its applications. We will, of course, take questions at the end, so please hold them then, and our speaker will be happy to address them. Globally, we're dealing with increasing medical complexities. The pandemic has highlighted the importance of zoonotic diseases, particularly with disease like COVID. We've also discovered long COVID or chronic and long-term effects from pathogenic diseases. Here's another example. Recently, Epstein-Barr virus was recognized as the major risk to develop multiple sclerosis. So chronic illnesses are another medical complexity to deal with in a rapidly aging global population. But aside from pathogens, our longer lifespans have introduced additional health challenges like cancer and dementia. So we require discovery of novel biomarkers and target validation in lockstep with development of effective treatment, essentially on demand. The IVD development pipeline also has to evolve. The next-generation protein sequencing is poised as an enabling technology across the whole IVD pipeline. Today's case study will focus on NGPS, primarily in the antibody discovery stages. But NGPS can be used in other parts of the IVD pipeline, and after our case study, we'll show ways that you can exploit NGPS and other forms of proteomics throughout the whole pipeline. It's my pleasure to hand things over to the stars of our show. Our guest speaker, Jijing Janet Wang, is a final year PhD candidate in the Zubarev lab in the Department of Medical Biophysics and Biochemistry at the Karolinska Instutet. Her PhD project, entitled Fighting IsoAspartate, a way to prevent Alzheimer's disease and maintain youth and health, focuses on elucidating the mechanisms behind IsoAsp formation from asparagine deamidation and aspartate isomerization, particularly as it relates to aging. She recently designed and developed an immunoassay targeting IsoAsp in human serum albumin to screen for Alzheimer's disease. Her work was recently published in Molecules in November 2021. Professor Roman Zubarev is one of the pioneers behind electron capture dissociation, ECD, which is routinely used in mass spectrometry analysis throughout the world. Research at the Zubarev laboratory centers on mass spectrometry-enabled molecular biometry to unravel the underpinnings of many cellular processes of biomedical importance. Monique Seymour graduated from the University of Glasgow with a master's in biotechnology and management. She enjoys working with scientists in both diagnostic and therapeutic areas to understand their research goals and ensure next-generation protein sequencing enables and/or complements current or new workflows to mitigate risk and deliver reproducible results. Monique is passionate about furthering life sciences research to improve global patient health. Janet, over to you.
Hello, everyone. Nice to meet you here. This is Jijing Janet Wang from Karolinska Institute, and my supervisor is Professor Roman Zubarev. First of all, I'd like to appreciate the invitation of Rapid Novor to let me share our research findings that we published last November, of which the title is first immunoassay for measuring isoaspartate in human serum albumin. Now, let me introduce the isoaspartate to you first. The IsoAsp is a damaging amino acid residue generated either from asparagine deamidation or aspartate isomerization, while the intermediate called succinimide. It often destroys native protein structure and function, and eventually initiates protein aggregation. Deamidation occurs in physiological conditions without enzymes, and it is also the most abundant post-translational modification in proteins. The deamidation rate could speed up as temperature and pH increase, and its accumulation has been proved to cause a series of negative effects in natural aging process, especially Alzheimer's disease and other neurodegenerative diseases. For instance, the isoaspartate accumulation in amyloid beta has been found to trigger AD. And our previous work also demonstrated the elevated levels of IsoAsp in the blood of AD patients. Thus, it's very important to test the role of IsoAsp in AD and other relevant diseases. So far, the quantification methods of IsoAsp have been focused on LC-MS with the electron transfer dissociation, ETD. The presence of IsoAsp could be identified by the Z8-57 ion, as shown here. But this method has two drawbacks. One is that it requires tryptase digestion of proteins ahead of analysis, which leads to the additional in vitro deamidation, and this could mask the biological differences. The other disadvantages of LC-MS are the low throughput and high analysis cost, while ELISA actually avoids both drawbacks. No need of digestion, high throughput, and scalable with low cost. However, such an ELISA requires a commercial antibody specific to a deamidated protein. So here come to our aims of this study. First, we'd like to obtain and characterize a monoclonal antibody against IsoAsp in human serum albumin, HSA. The reason why we chose HSA is that it's the most abundant protein in blood. Thus, we assume that the IsoAsp level in HSA reflects the general state of IsoAsp repair in organisms. Then we'd like to develop a simple and robust ELISA in quantification of IsoAsp in blood HSA and establish its normal range. Because of the large size of HSA, such an antibody had to be raised against a specific part of the HSA sequence. For that, we artificially deamidated the HSA protein by incubating it at higher temperature, 60 degrees Celsius, in alkaline condition, pH 8.5, for 7 to 28 days. The MS analysis revealed that the peptide LVNEVTEFAK exhibits the highest deamidation rate, with the IsoAsp occupancy reaching at around 63% after 28 days of deamidation. Furthermore, due to the exposed position of this peptide in the protein structure, it could be easily accessible by an antibody. Thus, this peptide was chosen to be the IsoAsp meter, representing the IsoAsp level in HSA. Against this peptide, two hybridoma clones were obtained, producing monoclonal antibodies 1A2 and 1A3. After comparisons of different ELISA methods, we chose the indirect ELISA using chemiluminescence for detection as our method, since it shows the highest signal-to-noise ratio at both 6% and 0.6% IsoAsp level. And this method is also the cheapest one. So here is the scheme of indirect ELISA. First, we coated the plates with fresh and aged HSA as antigens. Then we block it with 10% skim milk in PBST. Subsequently, the primary antibody and the secondary antibody were added respectively to the plate. Finally, the plate was detected by the chemiluminescence. From the figure here, we could tell that the monoclonal antibody 1A3 showed much higher specificity and sensitivity compared to 1A2, and the signal-to-noise ratio is above 3 already at 0.1% IsoAsp level. The DNA sequences of both 1A2 and 1A3 monoclonal antibodies were analyzed independently by two outsourcing services. The results are identical, and the corresponding amino acid sequences were 100% confirmed by another independent amino acid sequencing, done by ourselves and also validated by rapid novo. Only one amino acid in each of the light and heavy chains turned out to be different between 1A2 and 1A3 in variable regions. As shown in this slide, the serine in 1A2 heavy chain is replaced by isoleucine in 1A3 heavy chain. While in the light chain, only the serine in 1A2 is different from the leucine in 1A3. All the other amino acids are the same. Another information we got from amino acid sequencing is the glycosylation in both variable and constant regions. The figure shows the glycans attached in variable region of the light chain in 1A3. We could see from this box that the ones in position 28 and 30 are N-linked glycosylation, and the position 22, 26, and 34 are the O-linked glycosylation. We also characterized the binding between 1A3 and the aged HSA via surface plasmon resonance, which shows us the dissociation constant Kd is less than 10 to minus 6 molar, suggesting an above-average binding affinity. Besides, the hydrogen-deuterium exchange mass spectrometry, HDX-MS, also confirms the paratopes in the variable region of 1A3, which is right in the position where the amino acid is different from 1A2. In order to apply this MAB 1A3 in quantification of IsoAsp in human blood, we tested the effect of glycosylation on the specificity of the antibody, and found that after deglycosylation by 5 different enzymes, it made no more than 16% reduction of the 1A3 specificity. Thus, overall, the glycosylation doesn't affect 1A3 specificity dramatically. And then we generated another MAB based on the sequence information of variable regions in 1A3, but with a different isotype, the IgG2b, and we can find that the specificity of 1A3 star is still much higher than that of 1A2, while it is an order of magnitude below that of 1A3, so the isotype is also important. At the end of this project, we used the developed indirect ELISA to quantify the IsoAsp levels in HSA of 100 healthy plasma samples, based on the calibration curves generated from standard mixtures of the aged and the fresh HSA. The results fit the normal distribution with an average IsoAsp levels of 0.74%, using a linear calibration. Moreover, it demonstrates the good reproducibility as there's a significant correlation between two independent experiments done on two different dates. So to sum up, in this study, we characterized 1A3, the first monoclonal antibody targeting IsoAsp in HSA with high sensitivity and specificity, while 1A2 is not sensitive or specific enough to IsoAsp in HSA. And only one amino acid is different in both heavy chain and light chain of variable region between 1A3 and 1A2. The indirect ELISA using 1A3 as the primary antibody is feasible in measurement of IsoAsp in the blood HSA, with the advantages of high throughput and low cost. The normal range of IsoAsp in blood HSA is from 0.4 to 1.2%, with the average value of 0.74%. We published the paper on the Journal of Molecules last November, and apart from the 100 healthy donors that were studied in this paper, we've already tested the same assay in an AD cohort. The results revealed a promising potential of using IsoAsp in HSA as a blood biomarker in AD diagnostics. We hope this manuscript will soon be published online, and we believe it will be a significant application of this immunoassay I shared today, with meaningful values for the future AD studies. Last but not least, I'd like to express the acknowledgement to people and organizations that contributed to this study as shown here, concerning the fundings, resources, and technical support. And special thanks to Rapid Novo for the invitation to let me share with you today.
Thank you so much, Janet. Okay, Monique, your slide should be up, so take it away.
Thank you so much, Janet. It really is a pleasure to have you here with us. So, just a bit of background before I begin. Rapid Novo is a Canadian proteomics and next generation protein sequencing company that combines mass spectrometry with proprietary machine learning algorithms to enable researchers' next discoveries. As pioneers in the industry, we've successfully sequenced over 5,000 proteins and antibodies. So, needless to say, we're pretty good at what we do. Monoclonal sequencing was one of our first service offerings, which we continued to improve on, allowing us to sequence a wide variety of monoclonal antibodies from different species and isoforms. As we continued to develop, one of our new goals was to sequence polyclonal antibodies directly from serum. We have now successfully done this from both animals and humans, using both a proteomics approach as well as a proteogenomic approach. This has really opened the door for polyclonal sequencing to be used in early discovery processes, something that has generally not been explored before. Our drive to use de novo protein sequencing to enable research stems from our mission to decode immunity to better human health. We're fascinated by the immune system and all the proteomic interactions that naturally occur and that can be engineered to improve one's health. We currently are, and have been for a few years, the largest privately funded MS-based proteomics company in Canada. Our growth has been made possible by our team of hardworking scientists in tandem with the proprietary algorithms that were created by our founder and CSO, Bin Ma. As our company has grown, our partners and clients have grown in number and size as well. And we're really thrilled to be working with internationally renowned organizations such as the Karolinska Institute, contributing to such exciting research. So, just for some context, you might hear me say next generation protein sequencing or de novo protein sequencing. We use those terms interchangeably and sometimes just use the abbreviation of NGPS. So if you see or hear those terms, don't worry, They all refer to the same thing. Now, on the left-hand side, you can see a few images of some of the software and technology that we have in our lab. The top picture is an image of the peptide coverage we obtain when sequencing a protein. And we also have mass spec in there with some amino acid spectra. Now, our NGPS technology constitutes of REMAB, our monoclonal sequencing technology that I mentioned earlier, and REPAB, our polyclonal sequencing technology. These both can be used in antibody discovery-based research for IVDs as well as therapeutics. As a product of our monoclonal sequencing technology, we also offer protein sequencing of non-antibody proteins, which can be used for biomarker and biotherapeutic discovery. As we're also a proteomics-based company outside of NGPS, we do have a few other services that contribute to target validation and characterization of proteins. This includes MatchMap, which confirms the sequence of produced proteins. So, for example, if you'd like to validate that the protein you produced is in fact what you intended to produce, we have epitope mapping, which enables researchers to identify and analyze the binding regions of an antigen or binding protein. And we also have novo, which mines the immune response at different time points. Our proteomic solution can also contribute to pipeline integrated automations. So our service Novo Cloud also allows users to identify proteins. Now, I know I've mentioned antibody discovery previously, but our proteomic services can really be used in every stage of the IVD pipeline, as you can see in this image. Initially, in target discovery, MatchMab and Novo Cloud can be used earlier on in the process to help identify a target that could be of interest to researchers. For target validation, almost all of our services can be used to perform validation studies on a target of interest. So, if you're trying to confirm what the target is binding to, you could use Novo Cloud or MatchMab, or if you're trying to validate where on the protein it binds, you could use epitope mapping, and you could use REMAB to obtain antibody-based assays to validate the target. In regards to antibody discovery, our polyclonal sequencing service can be used to find an antibody. And Novo Cloud can be used to help identify antibodies of interest. In regards to diagnostic development, MatchMab can be used to ensure that the produced antibody or protein is what it's expected to be. And again, Novo Cloud's database can be used here as a reference. So this is a workflow of our monoclonal sequencing service. Everything is completed in-house by our lab technicians and mass spec specialists and our bioinformatics specialists. As I mentioned earlier, Rapid Novo is a highly quality-driven company. So every step that you see here is completed in such a way to ensure that we have the highest quality of data to drive our sequencing results. The REMAB platform is well established, and its workflow steps have been published in Clinical Cancer Research and the Journal of Proteome Research. Starting from a protein sample, it involves 4 basic steps of protein processing: liquid chromatography, tandem mass spectrometry, sequencing and assembly, and report delivery. Because everything is in-house, it's very easy for us to go back a few steps and run more tests in order to improve the results if needed. We would never release a sequence that we ourselves are not sure of. And what you also don't see in this diagram is that we offer post-project meetings to help guide you through the results. Although our primary service here is to provide the sequence, we also provide support and help your team figure out, what some next steps could be based on the research goal and the results. Our sample requirements are quite simple. We wanted to make sure that when sequencing, we don't require too much of the antibody, because there have been quite a few situations in which people have come to us with their last batch of antibodies they have and wanting to produce them recombinantly. So for that reason, we worked to make sure that we could complete the sequencing with 100 micrograms, and sometimes we can do it with less. In regards to purity, it's important for us to work with pure antibodies so that we can maximize the amount of good quality data that we obtain. If we throw a sample with lots of impurities through our mass spec, then we're naturally going to get a lot of noise. So for this reason, we ask for a sample of 80% purity in the standard buffer. If there is the presence of glycerol or BSA, this can make it more difficult, but we would just ask for more samples so that we can run purification steps. And as we've completed over 5,000 projects, we've basically been thrown every type of potential antibody our way. So we can confidently say that we can handle any species and any isotype. Whether it's a shark antibody or an IgM, it's no problem. As long as we know what we're dealing with, we're good to go. Now, one of the first things we do when we receive a sample is we tear it apart. So we don't even need the antibody to be functional when we receive it. We just need the full protein. When processing the sample, we use between 5 to 10 different enzymes. Our team likes to call it our protease cocktail. The reason why we use so many different enzymes is because it's created a higher number of varying peptides that contribute to the overlapping coverage of the sequence, providing a really holistic view of what we're looking at. Some of the protein chemistry and separation techniques, or our secret sauce, you could say, are patented and proprietary as well, which again contributes to the quality of data that we obtain. Now, once we've completed our digestions, we run the samples through our LC-MS equipment to get spectra reads on our peptides. Our mass specs also have the capability to do ETD-HCD, which stands for electron transfer dissociation, or high energy dissociation. This is an important tool as when it's paired with our machine learning algorithms, we can actually differentiate between isoleucine and leucine. We call this our WILD technology, which stands for W-ion isoleucine-leucine differentiation. Now, you might be asking, why is that important? Well, for those of you that may not know, isoleucine and leucine are isobaric, which makes it difficult to differentiate between the two. Without the use of WILDs, this poses a problem as depending on the number of isoleucine and leucines in your protein or antibody, this can create many potential variations. With WILDs, we can actually break the backbone of the two amino acids and differentiate the amino acids based on their side chains. Now, based off of Janet's results, antibody 1A2 and 1A3 differed by one amino acid in both the heavy and light chain variable regions. In the heavy chain, there was a difference of a serine to an isoleucine, and in the light chain, there was a difference of a serine to a leucine. Without WILD, this key differentiator could have been unconfirmed, as we wouldn't have been able to label the differences as isoleucine or leucine. And it's these key differences that really contributed to a change in the overall activity and specificity of the two antibodies in question. So after obtaining mass spectra of the peptides, we are then able to analyze the data with our proprietary machine learning algorithms. Although the machine learning algorithms are an integral part of our sequencing process, our bioinformatics specialists are always working closely with our software to ensure that the quality of data meets our standard. As I mentioned previously, everything is in-house. So at this point, if we're not happy with the coverage of the peptides that we have, we can very easily go back and run more tests to ensure that the sequencing and assembly are up to par. For context, in the industry, 20-fold coverage of the sequences is quite standard, and it provides about 95% confidence in the data. We strive for 30-fold coverage, which is 99-point-something% confidence. So after we're happy with the data, we combine everything in a report, which is then shared with the client. This report details the sequence of the whole protein, as well as calls out post-translational modifications that we find. We don't claim to find all of them, but things like cleaving or N or O-link glycosylations we do note down, especially if they're found on the variable region. Additionally, if there is the presence of extra heavy or light chains, which happens more than people ever expect, we'll also include the chains of that, and depending on the purity of the sample, we'll mention which one was found to be more actively dominant. We also share our coverage data with you, so you see the data that we found to convince ourselves of the sequence before sharing it with you. And again, based on the project, after the final report is completed, we can do further analysis with our other services or our partners like expression or epitope mapping or expression validation like MatchMab. So much like Janet's research, there are many other examples in literature of the use of NGPS's use to help enable research. In the case of APOE3, they sequenced the antibody to validate their discovered biomarker to help understand its role in Alzheimer's pathogenesis. And it also provides a framework for potential IVDs and therapeutics. In the second paper about abicimab, there is no cell or DNA input. So to understand its mechanism, there was no other way but to sequence the protein to adequately inform Cairo-EM and other structural analysis. It was found that the mechanism of action is mainly dominated by sterics. In the CAR-T case, they used our NGPS service to determine the sequence of a hamster antibody they had designed against 2 amino acid region that better allows for the purification of engineered T cells. Because they had a hamster, a not well annotated species, NGPS was the right choice for this project. In a different paper that cited our work, it was also discovered that thanks to our additional observations, the function of the neutralizing antibodies was dependent on sterics. NGPS is a great tool to uncover glycosylation sites, especially O-linked sites, which play a role in sterics, and again, which could impact the overall sensitivity of diagnostics. Overall, the key benefits of NGPS include not needing any cellular or nucleic input whatsoever, and working directly with the molecule of interest. This allows for minimal variation or interpretations, which may occur when working with DNA. Additionally, there is assurance of isobaric residues like isoleucine and leucine, ensuring that there is no potential variation of the data with the highest possible confidence. As we've seen with Janet's research and many others in literature, it can take one small change of an amino acid to have larger downstream effects on protein activity. So 100% surety is very important. Finally, with the protein sequence, you can directly inform structural analysis and better understand steric interactions for more robust development of diagnostics or therapeutic molecules and assays. In regards to in vitro diagnostic, NGPS can be used all throughout the diagnostic discovery pipeline, which can further enable sensitive and powerful amino standards for IVDs. Additionally, it also enables direct confirmation of the target by focusing on the actual protein of interest to truly understand function, ensuring nothing ends up lost in translation. And finally, because NGPS technology can be applied to any species and any antibody isotype, there are a wide variety of impactful benefits for both vet and human med to enable diagnostic development for overall improved health. So that's it for me. Janet, again, thank you and the Zubarev lab for joining us today. It was really interesting hearing about your work with developing an assay for Alzheimer's. And now I will open up the floor to questions.
All right. Thanks so much, Monique, and thanks again, Janet. Everyone, if you have a question today, you can certainly type it into the chat here, or of course into the Q&A feature. If you can't find those, try and hover your mouse over the top or the bottom of the screen, and we'll try to get to all of your questions in relatively short order. A very common question is, are the slides going to be available after the session? So I'll just preload that while everyone's thinking about the question that they want to ask. We don't typically make the slides available, but we are recording this session, so we should be able to share the link to the recording after the session. So with that, I'll try and start addressing the questions here. The first one from Sunny was nice work. I think this is addressed to Janet. So nice work. Does the specific IsoAsp residue also exist naturally? And if so, at what level in humans? So Sunny, I have to confess that we did a little bit of theater here. Janet's presentation here was recorded. She wasn't able to come on and do this live with us. But certainly I'll record your question and make sure that Janet gets that, and we'll get you an answer right back after the presentation. Sorry that we couldn't answer that one live. Vidal had a question about, can you also do this with highly purified peptide affinity polyclonal antibodies? Monique, do you want to try and tackle that one?
Yeah, thanks, Ian. We definitely can do sequencing with polyclonal antibodies. It is I would say a larger project, a bit much bigger undertaking, obviously, as there is much variation, but it is something that we have been doing for the past couple of years. And so I'm happy to talk about that further with you if you had a specific project in mind.
Very good, thanks, Monique. I don't see any other questions on the Q&A box, so we'll just give the others a chance to type in their questions before we wrap up. Oh, there is another one coming up here from Tim. So what are the ballpark costs for a polyclonal sequencing project? So I'll just take that one on, Tim. So we typically don't like to discuss pricing and costing for these sorts of things on webinars. You know, this is, I can give a huge order of magnitude thing. This is not in the hundreds or thousands of dollars, perhaps in the tens of thousands, certainly not in the hundreds of thousands of dollars, but it really depends on the exact nature of your project and what we're going to be trying to be doing. So again, that's going to be one for, I think, Monique to follow up with after the webinar finishes. Tim is also asking, thank you, is the wild procedure an add-on? Monique, would you like to take that one?
Yeah, thanks, Ian. The wild procedure is not an add-on. It's something that's integrated into what we do. So all of the protein sequencing that we conduct, naturally, we do determine the difference between isoleucine and leucine.
Yeah, and that's a really good question, because I think it was an add-on at one point. I think Rapid Novo introduced this capability. I think we were the first to do this as a commercial service, and when we first introduced it, it was an add-on, but now it's fully integrated. Looks like I've got one more from Tim. So does Rapid Novo use molecular cross-linking for epitope mapping to study engagement? Great question, Tim. I'd like to bring on an expert about HDX epitope mapping and cross-linking, perhaps on a future webinar, and we can go into a lot more detail on that technique. Got a question from Manoj. So the polyclonal sequencing projects are limited to protein targets, or can be done for small drug molecules as well? I don't have a quick answer for that one. Monique, do you have a quick answer from Manoj on that?
No, I don't either, but Manoj definitely put together some content for you and maybe discuss, what kind of small drug molecules you're thinking of. I think it's more of a loaded question that might have varying answers depending on your varying responses.
For sure. Yeah, there's a lot that can be done with that technology, I know. So that's a simple question with a complicated answer, I'm afraid. Hala asks, NGPS can deal with any type of species and isotype. Does that mean that we can do it with prokaryotic protein? And is it suitable for glycoprotein as well? Monique, again, I think that's a question for yourself.
Yep. So yeah, we can do prokaryotic proteins as long as, there's amino acids there making up a protein, we're good to go. And glycoproteins are something that we've worked with. We would just need to understand more of the nature of the proteins that it helps us guide how we approach the sequencing of it.
Yeah, and I think that's one of the advantages of the technique as well, is with proteomics being able to start looking at the other things that are going on aside from the primary structure. Would love to chat more with yourself, Hala, perhaps offline about the sorts of things that that you would like to see in your glycoproteins. Okay, we had several questions come in fast and furious there, and I think we've addressed them all. So probably it's time to wrap up. If you do have a last one that you want to get in there, get it in quickly. As I just wrap up with final comments here, so I wanted to say a big thanks to Monique and to Janet and the Zubarev lab. Thank you very much for joining us. Thank you all for joining us on this webcast, and thank you for your excellent engagement and wonderful questions. These webinars are always really great when we get lots of great questions and engagement from the audience. So thank you very much, and enjoy the rest of your day. And we hope to speak to you all soon.